I'd like to do for the first few minutes is actually take any specific questions that you heard, that you have from the two talks that you just heard. Yeah, Brad?
Fiona, I just have a question for you. You know, I'm just curious, I mean, has anybody actually, excuse me, has anybody actually done a kind of reverse experiment where they've contaminated water and actually seen how long the footprint remains after the fact? Because you're looking at a composite of everything that's been in that watershed, if you will, genomically. And I guess the question is just how long some of these DNA footprints that you're looking at persist.
I think that's a great question. Probably actually ask Natalie if she wants to comment as the person who's our lead sort of environmental person. But certainly, from my perspective, you know, we're dealing with just these very cases. Natalie, do you have any comments?
So this is a very, it's on, can you hear?
Yeah.
Okay. This is a very huge paradigm shift, actually going from the profile of the microbial community away from what we typically do in the drinking water community right now, is we focus on coliforms and indicators of fecal contamination, or we focus on pathogens specifically. So this is a major shift. We know a lot about how pathogens survive in water communities, and we know a little bit from aquatic ecology about some of these communities, but we're doing something very brand new. So we actually haven't done that reverse experiment where we take classic communities. The other thing is that the communities in freshwater aren't really particularly well characterized, so we'll be one of the first few groups who've actually characterized the freshwater microbiome from a public health perspective. I think there's 2 other groups in the US who are doing it, and that's all data that's available. And they're just going to start their projects as well, so we'll hopefully see some good data soon.
I think what's nice, though, is that we are looking at all microbes versus some of the past projects. There's been a lot of projects that have been looking at bacterial, for example, and not even focusing on viral. And as we know, there's a lot of phage, for example, that interact with bacteria and have a huge impact.
You have a question here?
Hi. My question is for Wyeth. I was just wondering what exactly your next steps are going to be with the promoter project. So you've got all of these promoters that are expressed in distinct regions of the brain, and it's some absolutely fabulous work that you've been doing, but I wasn't actually sure about the translation of that, like exactly what specific diseases you might be targeting with these promoters, or how you might sort of translate that into more practical applications.
So I'll give one example of one thing that we're working on right now. Now, in the long term, I'll just say that the diseases that we can go after in large part depend on the success that we have in terms of which cell types that we access with looking for. But right now, it's not exactly a brain disorder that we're getting in the first pass, because even more accessible than the brain to these systems is the eye. And so we're able to, with eye-based disorders, inject directly into the eye, and there's been some good success in that area. So we have a project right now related to aniridia, which is a disorder of mutation in the PAX6 gene, where the PAX6 genes are disrupted. And so what we're testing now is to see if we can develop promoters out of the PAX6 gene and direct expression of first a reporter gene in the eye, and over the next year and a half, also the PAX6 gene. And it's convenient because we have a mouse model of the system, and so we can observe whether we get it back. In the longer term, it's going to be sort of taking those promoters that emerge in the next year and a half, and then partnering with the clinical researchers who are of particular interest in those areas and going and working with it. The other side of that is that because we're partnered with the IKMC, these tools are being developed in parallel with their large-scale effort. And so globally, these promoters are going to be available for all mouse researchers who are working with the knockout collection.
So are most of these promoters, or most of these genes, are they disease-associated then, or?
Yeah, so yes, they're selected in large part by clinicians, and companies come to us and say we'd like to have an expression and certification. So for instance, we've had researchers say they'd like to get to the spiny neurons. They think that there's researchers say they'd really like to have something that expresses at the blood-brain barrier, so they could do that. So one of our promoters that we're working on is directed to the blood-brain barrier, and the idea being is if you could create a stable mechanism for transporting things across the barrier, it might open up for chemical treatments to the brain rather than genetic. So lots of little ones.
Thank you.
Any more questions at this point? I have a question for both of you. I'll start with Fiona. So one kind of nice thing with the shotgun data is that we are on this environment where we're able to identify the species that are there and create phylogeny of where they come from. But also as well, there's a huge amount of information in terms of the enzymatic activities that you're picking up. I was wondering, is it intellectual property or like a novel functionality that could be mined from these data too?
Yeah, I mean, it's a great point. I mean, my hypothesis is we're really going to see some significant changes in what bacteria obviously will suddenly increase in proportion in your samples for those that are deeply contaminated, suddenly you are able to metabolize all this material, right, and those bacteria will increase. And so you will see big shifts in gene function, proportions of genes of certain functions of these. And in fact, like I was trying to allude to, I think that will be more stable. But in terms of, methodologies, it's a huge sort of open area right now. There's a lot of very basic approaches being used right now that haven't, I think, really dealt with some of the artifacts that can occur. And obviously, in any bioinformatics analysis, I always tell my students, you have to watch out for these biases that we're just classifying things a certain way, and that will impact on how our analysis looks in the end. But at a recent microbiome session at a conference I was at, I mean, that was highlighted basically as a big sort of open area that needs a lot of research.
Yeah, huge amount of data. Yeah. Okay, thank you, Fiona. And Wyeth, just a quick question, because I obviously pointed out that when you make the promoters and you test them in mice, it's actually the human DNA that's actually being used to make the human promoters and the mouse promoters. So, were there any surprises in terms of moving a human system to a mouse to test, or is that giving you any insights into how conserved these processes are?
So it's a good question, and it's hard to make definitive statements, but I'll say a few things. One is we have to go back and look pretty closely to see if there was any evidence of differences between the human gene expression and the mouse gene expression for our candidates. And so when a gene comes forward, much of the data that we have available is from the mouse, Brain Atlas, the GenSat project, and these sort of large-scale mouse projects. And so you're basing much of your evidence from mouse genomic studies. And so you have to go back and look at the human expression data and say, is there evidence for the human gene being expressed in the same way? And it's certainly true that it's not uniform, but a portion of the time, wave my hands and say something like a third of the time, we see some differences between the human and the mouse gene and the expression that can be relevant. So, yes, it's a challenge in that way. Now, in terms of what we get out, I can give you the general thing. So when we looked at our Pleiades project and we said how many of our designs directed expression, it was about, I'm going to say it's about 25% of our design, 20% of our designs. Of those, about half of them gave us the expression that we were targeting in some form, that they were overlapping with the endogenous expression. And about half the time that we were getting expression off of them, it was going to the brain, but it wasn't to the region that we expect them to be. So we don't hit it perfectly.
Okay, thanks, Ryan. There are no other questions, but I'd like to just kind of move to the next part of this session. And Fiona and Wyeth were chosen to speak something not just because of the two projects that they talked about, but because they're very accomplished bioinformaticians in their own right and working here in Vancouver and Burnaby. And there's a great deal of interest, particularly now around the world, and particularly in Genome and Canada as well, in terms of looking towards bioinformatics even more to solve many of the problems. Particularly now the next generation DNA sequencing has given us even more computational and investigational challenges to undertake. And concept is that a lot of the innovation that's required to move us forward is we're relying on bioinformatics to come up with that. So what I did, I've come up with a number of kind of questions to ask both Fiona and Wyeth to give us an idea of where they think the field is going and what we think we need, what they think the field needs to do, and what the potential is of the field. So I'll start with my question, if I may, you two. So my first question, and it's partly motivated by Fiona's talk, which is we're doing research, but we want to actually apply it into the field. And so the question is, what are the differences in the demands of bioinformatic applications when they're actually used in the clinic or for environmental health and away from a research environment? And do we have the bioinformatics infrastructure in Canada to undertake and deliver such applied uses? So I guess we'll start with Fiona.
I have to say thanks to Steve for actually giving us these questions ahead of time so we have time to think about it. And I also have to acknowledge Ryan Brinkman, my husband, who I asked to get a nice conversation over wine last night over some of these issues. But I will say that Ryan and I both agree that in terms of translating bioinformatics into the clinic, high accuracy and ease of use are absolutely critical. And I really liked actually Karsten's comments about differentiating clinical informatics from bioinformatics and the importance of coming up with bioinformatics or almost sort of clinical informatics support in the clinic that is really much more geared to be usable in a clinic versus, say, bioinformatics being done by bioinformaticians. I think it's an open question about to what degree we're actually going to get bioinformaticists working in very applied clinical environments, or you could translate this all, of course, to working in Ministry of Environment on environmental issues. Certainly, I see sort of bigger institutes having sort of bioinformatics that have research support. We'll have more research-oriented projects. But for your little clinic that doesn't have that kind of research focus, I think it's a huge challenge, in fact, right now, that we need to have training of how to get people to be able to make their software be suitable for that translation. We have a lot of training in bioinformatics for algorithms and doing appropriate, effective methods. But then we need to get it to be then we need to take another step and train people on how to make these methods, social engineering, to how to make these methods be easy to use and accurate in the clinic.
So this becomes a huge challenge in the bioinformatics community as we move to become clinical informaticists, in many ways we're being forced to because of the dearth of skills in that area in the community. So being based at Easton Children's Hospital, we see that intersection arising a lot in our research of late. And so, for instance, we have a large-scale project where kids are coming into the biochemical diseases clinic. It's pretty clear that they have a mutation in some important metabolic enzyme, but they don't know which one it is. And so they start tracking them down and trying to map them out. But about 30% of the time, they know no one to go after, and they just can't figure out what's going on. So the natural extension, as you heard this morning, is to go out and perhaps sequence through them and do exome analysis and genome analysis. And so we get pulled into that, and so it's a research endeavor at this point. It's not really clinical delivery. But at the research side, we can really see where we have all sorts of challenges in terms of the interpretation, quality of the data. You're hiding that all under the hood to try to make it interpretable to somebody at the front lines. And you're finding that while the cost of doing the actual sequencing might be coming down to under $1,000 now per sample, the time spent and the cost spent on the informatics and the interpretation of the data is closer to $5,000 or $6,000. And no one wants to budget in $5,000 or $6,000 for interpretation and staff time to understand the genomes that they come back. They think it's just going to give a nice list and they're going to read it off. We all read these beautiful papers that say everything's clear cut, but you have to realize that 2/3 or 3/4 of the cases that have been large-scale sequencing applied, it hasn't been clear cut, it never makes it into the journals, and that there's been a ton of time spent